Hi everyone, welcome to this video lecture about proteins. Proteins are one of those large biological molecules called macromolecules, along with carbohydrates, lipids, and nucleic acids. But we are going to be focusing on proteins in this video. And I do think proteins are one of the best macromolecules. And one of the reasons for that is that they just do so many different things. Pretty much anything that you can think of within an organism, any process, any function, any characteristic has at least something to do with a protein. So, proteins do a ton of things, and this slide is dedicated to sort of giving an overview of some of those functions. So, one of the things that they do is they have to do with making us who we are physically. Proteins play a big role in the structure and sort of basic features of an organism. So, if you're thinking about your hair and your fingernails, those are made of proteins. The color of your eyes, the color of your hair, that has to do with pigment proteins that are causing your eyes and hair to be the color that they are. Also, if you think about movement, so things like muscles, cartilage, bones, tendons, all of those are structural pieces that allow us to exist and get around. Those are all proteins. There are a bunch of other functions, storage, especially things for like feeding young, nutrient-rich things like egg yolks. All of the chemical reactions in our bodies, the chemical reactions that have to do with photosynthesis or cellular respiration, those all need to happen more quickly than they would happen on their own. So these special proteins called enzymes are responsible for speeding up those chemical reactions. Proteins also have to do with communication, so things like hormones, receptors for different signaling molecules. All of those are important for this says communication, communication roles, defense, so things in your immune system um, called immunoglobulins, and then as well as transport. So hemoglobin is a molecule that carries oxygen around. We have channel proteins in our cell membranes that allow large or charged molecules to get in and out. So, hopefully, this gives you a picture or a sense of all the different kinds of things that proteins do for us. So, this slide is about the structure of amino acids. And amino acids are the subunit or the monomer that make up proteins. When you get down to the most basic level or most basic kind of structure for a protein, it's essentially a long string of these amino acids. There are 20 different kinds, and if you put them together, sort of like beads on a string, like a necklace, or if you think about them like little building blocks that you can attach to one another, essentially putting together these 20 different amino acids in different ways are what give us proteins. So these amino acids have, like I said, there are 20 different kinds, but a lot of their structure is identical for all the different amino acids. When you look at any given amino acid, there are three main parts, and two of those parts are identical, and one part is different between different amino acids. So the identical parts are this amino group. So that's shown here in blue, nitrogen and three hydrogens, and then a carboxyl group as well. So this is a carbon and two oxygens. So this top half, everything except for this R that's highlighted in yellow, that's all the same for every single amino acid. So all these 20 different kinds, they all have this top half identical. The part that's different between the different amino acids, the part that gives us these 20 different kinds, is this R group at the bottom. So basically, where this yellow box is can be a bunch of different kinds of things. And you can see that here. Basically, all the stuff that's highlighted in green here, the stuff that's highlighted in pink here, in this rust color here or blue here, you can see that that's all different chemical structures for this bottom section down here. And that different R group is what gives these different amino acids different kinds of properties. So some are going to be hydrophobic, some are going to be hydrophilic, some are positively charged or negatively charged. These different R groups are going to give each amino acid their special properties. We form amino acids, like I said, by linking up. We form proteins, like I said, by linking up amino acids. So we join together chains of amino acids, and when we join together multiple amino acids in a chain, we call that chain a polypeptide. And there's a special kind of chemical chemical reaction called a dehydration reaction, where basically we're taking out a water molecule. So you can see two H's here and an O here. That makes water, H2O. So we take that out, and in that process, we join together these two amino acids. So if we do this process many, 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 many times, we can get a long chain of amino acids to form. So these long chains of 20 different amino acids result in proteins being able to take on so many different jobs and shapes. That's because proteins or polypeptides are typically about 50 to 2,000 amino acids long. So that's quite they can reach quite long lengths. And like I said, we have those 20 different kinds of amino acids. So this is different than when we talked about carbohydrates and things like polysaccharides, because polysaccharides are also long chains, but there was only one subunit, only one component that we're using to build those chains. So when we're thinking about things like starch or glycogen, those are also long chains, but they were made up of just one thing, just all glucose molecules one after another. So just a bunch of glucose molecules chained together with slightly different branching patterns. But since we have these 20 different amino acids, we can create um, millions of different combinations based on the specific amino acids that we have and the order that we put them in. So, this allows proteins to have so many different shapes and chemical properties, and that's what allows them to have such varied functions. Okay, so the next thing that we're going to talk about is how proteins get their shapes. So, there are four levels of protein structure. The primary level of protein structure, the first level, is just the sequence or chain of amino acids. So, what amino acids that protein has and in what order they come in. So, that's primary structure. So, just no folding, just the long chain of amino acids. Secondary structure has to do with folding up that chain into some sort of local structures. And so the two most common forms of secondary structure are sort of curlicues, and we call those alpha helices, and then also sort of pleated or zigzag shaped. We call those pleated sheets. So secondary structure is just when we take that chain of amino acids and we start folding them into these repeating patterns. Tertiary structure is the third level of structure, and that's when we fold these zigzags and these curlicues up upon themselves. So if you're looking here, you can see this zigzag here. That's a pleated sheet. There's an alpha helix down here, a curlicue right here. So we're just further folding up that chain of amino acids even more and putting different regions of those chains close together. And then the final level of structure is called quaternary structure. And this has to do when multiple chains of amino acids come together. So multiple subunits combining. And so one example of this is hemoglobin, which carries oxygen around. It actually needs four different subunits in order to be fully functional. So I just want to re-emphasize that proteins have these very complicated three-dimensional shapes, and those shapes are ultimately determined by the amino acid sequence or its primary structure. And having the right shape is essential for that protein to do its job properly. If a protein becomes folded incorrectly or becomes unfolded, then it can't do its job and it's kind of completely useless. So this last slide has to do with protein folding, and I want to introduce this term of denaturing. So if you denature a protein, that means that the protein has lost its proper shape. So uh, if we denature a protein, you can see a picture here, and when it's in its active form and it's doing its job, if it gets unfolded, then it's going to not be doing its job. And sometimes you can see that visually. So, for example, if you've ever cooked an egg, you've seen that the egg white and the egg yolk sort of change color. The egg yolk or the egg white starts as being translucent and it turns white. And that's visually what you're seeing is those proteins in the egg white denaturing. So you can cause denaturation by heat. You can do it with changes in pH. You can do it by physically sort of like agitating the protein, sort of moving it around too much. Denaturation can be permanent, so it's kind of impossible to unscramble an egg. but it can also be temporary. You actually denature proteins if you warm up milk, but if you let it cool back down again, the proteins can take on their proper shape, uh, retake on their proper shape. So it can be permanent, it can be temporary. But the main thing is that if the protein doesn't have the right shape, it can't do its job. So denaturing proteins is detrimental to their functionality. Okay, so that's this lecture for an introduction to proteins, and I will see you all soon.